Yes, my dears, we were talking about the bacterial structures and bacterial size and shape को हमने discuss किया है. Let's see the structure of a generalized bacterial cell or a prokaryotic cell. First of all, we can create a concept map for studying this very small creature, and that concept map is that we can divide whole of the cell into three portions or three regions. One is the external structure. External structures are very necessary for bacterial cell because it is very small living being and it has cell wall around it, and you know that cell wall is a dead material. So for getting the stimuli and response of those stimuli, there is need of some other structures which are external structures. So external structures include flagella, pili, and Glycocalyx. Pili are also sometimes used by some biologists as fimbria or fimbri. Fimbria and fimbri. They can be used interchangeably, and most of the biologists use these structures interchangeably. And then Glycocalyx. Glycocalyx is further divided into slime layer and capsule. Then the second part of the bacterial cell is envelope, and this envelope consists of two structures. One is the cell wall and other is the cell membrane. Then there is the third portion of the cell, and which is cytoplasm. Now cytoplasm of the cell, a bacterial cell, can be divided into two. One is the cytosol, soluble part of the cytoplasm, and then internal structures are cell internal inclusions. These includes the nucleoid. You all know what is a nucleoid. It is just like the nucleus, not nucleus, but just like the nucleus. It is the region inside the cytoplasm where bacterial genome is present, bacterial DNA is present. Then the ribosomes of bacteria are also present inside the cytoplasm. And another very important structure, plasmids, are also there, which are containing the disease antibiotic resistant genes, are genes for other purposes, and they can also be used as vector in biotechnology and genetic engineering. Then we will talk about cytoskeleton and some other cell inclusions. So these are three portions of the bacterial cell. So we can discuss them one by one. Let's first of all start external structures. In external structure, the most important and the prime most is the pili. Pili is the plural of pilus, and pilus mean hair-like structure. These can be seen with electron microscope, and very fine structures, pili, can be seen, and pili are most oftenly present in gram-negative bacteria. Pili are very important structures because they allow the cells to attach the surfaces of other cell and surfaces of other substratum. These pili are very specific, keep it in mind. Just like the receptor site in case of viruses, अभी हमने recently पढ़ा है कि viruses के जो attachment fibers हैं, they can attach on the host cell only where they have receptive sites. Here the pili are also very specific, and pili of different bacteria are specific for different kind of cells. For example, pili of Neisseria gonorrhea, which causes gonorrhea and a disease, you have already studied it, urinogenital problem. Neisseria gonorrhoeae pili can attach only to the urinogenital cells, and Bacterial pertussis, which causes whooping cough, its pili can attach to the upper respiratory tract cell. So it means that they are very, very important. Pili not only help the cell to attach the other cell and help the cell to attach to the other surfaces. There are some bacteria which produce specific pili, which are conjugation pili. And you know that bacteria exchange their genetic material. There is exchange of genetic material among bacteria because of conjugation pili. So conjugation is one of the method of recombination in bacteria. Other will be discussed later. You know what is conjugation, and they produce conjugation pili, and they form a conjugation tube between two cells of two bacterial cells. So pili made up of pilin protein. They are named so because their hair-like structure can be seen in transmission electron microscope, and they are very specific. Neisseria's pili can attach the urinogenital tract, and Bacterial pili can attach to the upper respiratory tract cell. And conjugation pili, they will attach to the conjugant to the other complementary cell of the conjugation. I hope I should let you see. Here is the structure of a bacterial cell under transmission electron microscope. It is transmission electron micrograph of Escherichia coli, one of the famous bacterial cell. You can see these green hair-like structures. These all are pili, and this blue structure is definitely, this one is the Escherichia coli, the bacterial cell. So let's move forward. Flagella. Flagella are also very important structures. Their number vary in different cells. I wanna let you see. These are 3 flagella you can see on a bacillus. एक bacillus के ऊपर आप ये 3 flagella देख सकते हैं. So flagella, their number varies from 1 to many. Monotrichous, if you remember, हमने कहा था if there is one flagellum, it is monotrichous. If there is no flagellum, atrichous, and lophotrichous, and amphitrichous, and peritrichous. I hope you remember all these terms on the basis of the number of flagella and their arrangement of the bacterial cell. Size of the flagella varies from 10 to 20 micrometer in length. They can be more than their body length. A flagellum consists of 3 parts. एक flagellum के 3 हिस्से होते हैं. One is the basal body, very complex structure, and the other is the hook, and the third one is the filament. So there are 3 basic parts of the flagellum: a basal body, a hook, and filament. Flagella are actually the motility structures. They enable the cell to be motile. ये सेल को मूव करने में, लोकोमोट करने में हेल्प करते हैं, and their locomotion is very interesting and astonishing rather. The flagellum rotate counterclockwise, and it not only help in the movement of the cell, rather it also help in the temporal sensing. It is also one of the, you may say it is one of the receptor structure of the cell. ये सेल का receptor structure है for receiving the chemical and other stimuli in the environment. So their flagella rotate counterclockwise. Cell move in bursts, which are called runs. You might be knowing the runs. Have you heard the word runs in cricket? You know, players move very fast among the in between the wheels, and this is called the run. Here the burst of the cells are actually the runs. ये runs कहलाएंगे, and it is because of flagella. ये फ्लैजेला की वजह से है. Flagella's internal structures, you all have studied that there is flagellum protein and protein fibers are there. There is very intricate and interesting arrangement of the protein fibers inside the flagella, which make it possible the counter clockwise rotation. The third part of the external structure are the glycocalyx. Glycocalyx. It is also very important structure. What is glycocalyx? Glyco means sugars, sweet, meetha, and calyx means the cover, protection layer or protective layer. So it is glyco means polysaccharide or carbohydrates and calyx cover. So it is one of the cover of the cell. It is around the cell. Suppose this is the cell and this is the cover of the carbohydrates. Carbohydrates are covering the cell. So it is glycocalyx. Glycocalyx is if it forms very thin layer of dextrin. Dextrin, you might have studied this chemical structure, biochemical or biomolecule, dextrin. And if there are very thin fibers of dextrin around the cell, then it is called slime layer. फिर तो हम इसको slime layer कहते हैं. But if it is very thicker and there is a layer of carbohydrates along with some small amount of protein, then it will be named as capsule. It means the glycocalyx can be of two types. It may be a capsule if it is a thicker layer of carbohydrate along with small amount of protein. So capsule is actually carbohydrates plus small amount of proteins. When it is present, it will be named as capsule, and if it is a thin layer of dextrin fiber, then it will be slime layer. Both of these structure, for example, it also help in the attachment. This glycocalyx, those bacteria which do not have pili or flagella, their glycocalyx help them to attach to their host cell or the host tissue. They play the role of a buffer between the environment and the cytoplasm. Cytoplasm and environment के बीच में एक buffer का role अदा करते हैं. You know what is buffer. They also help the cell, bacterial cell, to avoid desiccation. कहीं evaporation से bacterial cell में से बहुत सारा water loss ना हो जाए, इससे भी ये रोकते हैं. They also help in the attachment of the cell to the host tissue or cells, and they also help in the virulence. Those bacteria which have capsule or slime layer, they are more virulent as compared to others. What other structures are there? Envelope and cytoplasm we will discuss later. I think you understand. If you have any problem, you can ask me the question, my dears. Thank you very much. Allah Hafiz.